Yep, okay, so we'll start with that. So just kind of telling you about a little bit about the background of this study, of why we've been working on this concept for the past actually several years now, and why we really thought it was extremely important. So treating or doing appropriate clinical trials, especially in the US, for patients who are elderly, frail, or some categories of intermediate fit patients, that is still an unmet need. There are hardly any clinical trials. In fact, I can say maybe we can count the trials dedicated to this population in the US in multiple myeloma maybe on 1 or 2 or 3 maybe fingers. That's about it. This patient population has always been left out of clinical trials. We also know that historically there had been a regimen called VRD Light, which has been used very frequently for this patient population. And then subsequently, there is the regimen of daratumumab with lenalidomide and dex, so DRD. That is FDA approved, and based on this one clinical trial, Maya, which was done primarily in transplant ineligible, but I should also say that not all the patients were necessarily frail or elderly, because historically these terms have been used somewhat overlapping. We've also mentioned that patients who meet this elderly frail criteria tend to not get enrolled in prospective clinical trials frequently. And our hope was to create a study that was as close to real-world population and real-world use as possible. We also wanted to introduce some practical study inclusion criteria. So we wanted to test things, a concept that has not been done before, at least not in myeloma studies, where the inclusion criteria could be modified. And I'll go over all of those in a bit detail in a bit. So this slide tells the study schema, which is basically it is frail or a selected subset of intermediate fit patients who are newly diagnosed with myeloma would be randomized to these 3 arms. From the arm standpoint, I want you to look at both the induction and the maintenance, because that is how the arms are set up. Arm 1 is the VRD Light, followed by Revlimid or lenalidomide maintenance. Arm 2 is DRD followed by Len maintenance, and Arm 3 is DRD followed by Dara-Len combination maintenance. The rationale for that is because while the Maya trial that brought DRD to FDA approval has the full regimen to be used kind of till progression or side effects, but in the real world we've seen, and there's data published out of real world analyses, that in real-world use, daratumumab lenalidomide gets given for the first 6-8 months, and then the regimen goes to a quote-unquote maintenance term, which is an unofficial term used, and the regimen is frequently just brought to single-agent lenalidomide. So the Arm 2 is mimicking the current real-world usage. Arm 3 is the recommended DRD usage, and Arm 1 is the VRD Light. Patients would be treated until disease progression or until there is any adverse event that requires them to come off. These are some stratification factors: frail versus intermediate fit as classified by the Myeloma Frailty Index, presence of 1 or more of the high-risk cytogenetics. It's deletion 17p, 1416, 1420, 1q+, or 414. And if the patient got any systemic anti-myeloma therapy before, I will explain the protocol allows for a little bit of treatment to be given before study entry, just to again accommodate patient enrollment and facilitate that. The study objectives are a little, this is again a little bit different way of looking at it. As you can see, as you noted in the schema, the induction and maintenance, both of them make up the arm. The maintenance is common between Arm 1 and Arm 2, and the induction is common between Arm 2 and Arm 3. So it's hard to pick up just the PFS or just the OS and compare all 3 arms. So the study was set up with a dual primary objective, which would be to compare the progression-free survival between Arm 1 and Arm 2 and the overall survival between Arm 1 and Arm 3. So either one of these coming back as positive would report the study to be a success or a positive study. So progression-free survival comparison between Arm 1 and 2, or OS between Arm 1 and 3. A bunch of secondary objectives looking at the other analyses, progression-free survival, overall survival. Eventually, everything gets compared to everything. And then safety, then median time to response, to best response across the 3 study arms. And we are going to be exploring the venous and arterial thromboembolism, so VTE risk, which is being set up as a sub-study or a sub-analysis. We also have some quality of life objectives that are baked into the protocol. So these have been approved as part of the protocol because we were very, very interested that this would be extremely meaningful for this population. And so the quality of life was set up prospectively. We didn't necessarily want to go and retrofit the study and miss out the opportunity to collect QOL in all the patients. So that is covering this EORTC questionnaire and the QLQ-C30 questionnaire and a couple of patient-reported CTCAE criteria of fatigue, peripheral neuropathy, and pain. We also have MRD assessment as an additional objective, but I should mention that the MRD assessment is being done in this study as standard of care. This has been discussed on several meetings with the SWOG community across this meeting, calls, open session, closed session, and it was considered that by the time the study activated, which was this year, MRD assessment by clonoSEQ analysis is considered a standard of care test, and so we should be able to do it as part of that. Of course, this is not a part of it is not an integrated endpoint, meaning that we would like this to be collected as much as possible and be tested at the appropriate time points, which is at the end of 9 cycles of induction. And then it will also be after 1 year of maintenance. So I think the schema didn't maybe clarify this fully, but the patients who start on any of these arms get 9 cycles of induction on each of the 3 arms, and then they go to maintenance, and the maintenance continues till progression or any adverse event. So the MRD assessment is at the end of induction, at the end of 1 year of maintenance, and then those are the dedicated time points that we have said would be extremely important. And of course, it's the conversion and then sustained MRD negativity. Eligibility criteria. So some of the standard eligibility criteria, for example, the measurable disease, but you will notice that patients are allowed based on labs within 28 days prior to registration, as most of our templates are 14 days prior to registration, but we lengthened that time just to make it a little bit easier for patients to get in. Patients who have IgA, IgM, IgD, or IgE type myeloma, a lower M spike is allowed, going with the fact that these tend to have lower secretion. Light chain myeloma, of course, allowed. Patients will be allowed, they will not be allowed any prior systemic therapy, but the exceptions are using a short course of corticosteroids for palliative purposes of symptoms is allowed, as long as they get up to 1 complete cycle of a non-daratumumab, non-Darzalex regimen. So if a patient gets a doublet or even a triplet for that matter, but as long as they have not had daratumumab for a full 1 cycle. Well, the caveat you can imagine is that if the patient gets a couple of doses, less than 4 doses of the daratumumab, they would still be able to qualify because they have not had 1 complete cycle of a daratumumab-based regimen. Localized palliative radiation therapy is allowed, but it must be completed at least 3 days prior to starting the study treatment. These are again some flexibility that has been added because generally in our templates it has been at least 7 days or 14 days prior to starting treatment, radiation has to be finished. So we've brought that closer. This frailty score or calculator is embedded in the protocol. I would highly recommend those sites who are activating the study, please maybe add this to your favorites or on the websites and the computers, just because it'll be easier to go to. This calculator helps calculate the frailty score. Patients who are frail, you will notice that we are giving a high, the hemoglobin allowance, for example, in our protocols typically it's above 8. We're allowing above 7. Platelets above 50 are being allowed. And the ANC, you will notice typically the allowance is ANC of 1. We're allowing even if the ANC is at 0.75. And on top of that, growth factor and transfusion utilization will be allowed if it is considered by the PI, that the treating physician, that the cytopenias are secondary to the disease. Bottom line is, these are available regimens. If the patient was to come to our clinics, they would get these regimens anyways. We would like to facilitate that that patient gets the treatment on the trial rather than getting the same treatments as standard of care. So we are making it easier for a patient to get onto the trial and for the physician to be able to get the patient on the study. Now, if the patient scores intermediate fit, then you will notice, and I should also say for the frail patients, any degree of kidney dysfunction is allowed short of dialysis. So the patient can have a very low creatinine clearance, less than 15, whatever. As long as they are not in dialysis, they can go on. There is no limit of kidney dysfunction. If the patient calculates as intermediate fit, then the eligibility criteria are a little bit different. The patient's hemoglobin has to be between 7 or 8, as in below 7 is not allowed anyways. Above 8, no. So there is a smaller window in which the patient has to land if they are intermediate fit. And I can explain the background of that. Platelets between 50 and 75, and ANC between 0.75 and 1. Or if the patient is intermediate fit and has RISS stage 3 disease, or if they have creatinine clearance less than 30. The reason for all of this is because for patients who have intermediate fit creatinine clearance above 30, intermediate fit hemoglobin above 8, intermediate fit platelets above 75, or intermediate fit ANC above 1, or intermediate fit RISS stage 1 or 2, there is a current ongoing clinical trial that those patients fit into. So a strong thought was that we did not want one cooperative group study to be competing with a second cooperative group study. Now, as that other study completes its accrual, if there is a possibility, we can see whether or not this needs to be modified, but we have to make sure that that should not affect the study's integrity and the stats, etc. But for now, if a patient is intermediate fit, this has caused some questions from sites. They have asked us to explain. This is a little tricky. We are, as study chairs, available to answer any questions anytime, but I just want everybody to please make a note of this. The fact that frail patients, I'm spending a little bit extra time on this, frail patients, very broad criteria. Intermediate fit, a very narrow range, okay? Any lenalidomide dose modifications for kidney dysfunction should be done as per the treating physician based on standard criteria. The antithrombotic prophylaxis, so anti-blood clot prophylaxis or herpes zoster prophylaxis should be standard of care. That is with IMiDs or with bortezomib. Pre-medication supportive care, et cetera, institution guidelines. The protocol is not mandating any of that supportive care because these are FDA-approved standard regimens. And the concomitant medications again, et cetera, as per the treating physician's assessment because these are standard regimens. These are the regimens and schedules. You'll notice that bortezomib is given subcutaneously and weekly. This is in tune with the VRD light regimen. A lot of the bortezomib-based studies have still used twice-a-week regimen, but this is, again, going with the VRD light weekly. Lenalidomide is 15 milligrams against the standard starting dose of 25. This is for frail elderly, so 15. And the dexamethasone is 20 milligram weekly. So this is the VRD light regimen. And then lenalidomide goes to 10 milligram on maintenance, starting at cycle 10. Each cycle is 28 days. Again, something to keep in mind, VRD is sometimes given as 21-day cycles. This is 28 days, again going with VRD light. Arm 2, DRD going to Len. The Darzalex is standard of care. Lenalidomide is again 15, and Dex is again 20. Maintenance is Len 10. And Arm 3, the induction is the same, but over here, the maintenance is with Darzalex and lenalidomide. Darzalex is monthly. We want to say that we are not anticipating any side effects that are different. These regimens have been studied independently in this population, so nothing that we are considering to be an adverse event of interest. But we do want to point out to all the treating physician sites, et cetera, that this is a special population, so I think a little bit more kind of focus on the side effect profile. So be cognizant of the fact, I'm pretty sure all of us who treat myeloma have used these regimens in this exact same patient population. But because we are allowing for a little bit, the labs, et cetera, are a little bit lax, slightly broader, so considering side effects would be important. This slide goes over dose modifications and interruptions. This is some general guidelines. For example, missed doses will not be made up. If a cycle is delayed, maximum delay, dose delay for any reason would be 8 weeks. Multiple toxicities happening, then the dose modification should be based on whichever toxicity requires the largest dose reduction, and the physician will have to assess which toxicity is coming from which drug. Once a dose is reduced, participants will continue at the new dose level. We're not asking to escalate again specifically because this is a special population. We're also saying we're allowing any degree of kidney dysfunction, for example. So things to just kind of keep in mind there. No dose reductions for the Darzalex itself. Again, talking more about this is more so going with the package insert, but frankly, these guidelines of daratumumab dose modifications are literally taken from the package insert. So this is in line with the standard of care usage of this drug. Again, I'll skip over this particular slide because this is again talking about those same standard guidelines. These are some, the slide 19 is talking about some dose modifications and interruptions of len and bortezomib. Again, very standard, going by the package insert. Nothing that is being done specifically or especially for this study that is different from standard of care practice. Again, dose modifications, and you can see obviously there is so much of this regulatory language that is required to be disseminated, and hence all these extremely dense slides which you can review, but from my standpoint, extremely important to keep in mind, this is going with the standard of care practice. And there are some dose modifications or interruptions that we do recommend or have pointed out for dexamethasone also. They are listed in the protocol, but I can again tell you that's very standard of care process that we use. Now, criteria for removal from study, of course, unacceptable toxicity. If the patient has disease progression, then certainly they would come off. If there is a treatment delay for any reason of more than 8 weeks consecutively for daratumumab, or there is a, so if there are delays, and if there is any question, actually I would say forget about even the delays. If there are any questions, we as study chairs are available, and actually the SWOG, the whole of the operations team is excellent in helping troubleshoot a lot of things. So we are available to answer any questions. And of course, participants, of course, may withdraw from the study at any time if they want to. Site initiation and participant registration, these are all very standard processes, but links to all of them are provided in the protocol documents. Of course, since the patients are going on lenalidomide, they will have to be enrolled on the REMS program, but that is again in line with standard practice. Now, daratumumab is being provided by the study. So this, although I keep saying that this is standard of care regimens, the Darzalex is being provided by the clinical trial. So there is a specific drug ordering process for arms 2 and 3. So bortezomib, len, dex on all the arms are standard of care commercial supply, but the Darzalex comes as a part of the study. So there are ordering guidelines. Study assessments, I think we'll go over a few of these, but what I'm going to point out is only some of the specific ones that we think are going to contribute to the objectives of the trial. So PET CT scans, for example, we do want them at baseline. We do want them at the end of induction therapy to assess the response. Then at the end of 1 year of maintenance, again to assess the response. And at any time there is concern of disease progression or it's clinically indicated, of course it can be done. PET scans are being done as standard of care, and we've spent quite a bit of time carefully assessing that if they are done for response assessment or for restaging purposes, then of course they should be covered as standard of care. So PET scans are to be done as standard of care, but we do considered that doing them at these specific time points will help kind of standardize the data collection. Data submission is, of course, through RAVE, Metadata RAVE. The master form set is accessible through this link. The link works because I did download them and review the forms. It was obviously fine. The PRO questionnaires are also accessible because there are those 2 questionnaires that we really want to be done at the right times. Data submission guidelines are all standard, nothing specific that is being done specifically for this particular protocol. Now, there are some specimen submission guidelines and time points. So at these particular time points, which is baseline prior to starting treatment, end of induction, which is following cycle 9, and then end of 1 year of maintenance or disease progression, whichever occurs earlier. At those time points, these bone marrow aspirates, bone marrow biopsies, and peripheral blood samples are to be submitted. We have listed over here the how many tubes, how much quantity, etc. And in fact, just today there was a question that came from one of the sites asking some clarification on this. I think they just asked, what if the site is not ready to release a block? Because certain sites may have some restrictions. So we were able to find clarification from the tissue bank, and while we have submitted or given that information right away to that particular site, we do plan that in a subsequent amendment, we will clarify that portion. As I mentioned, the specimen submission is extremely important. We do hope to be able to generate a lot of important analyses out of that through the correlative science. The specimens will all be shipped to the SWOG biospecimen bank. I should again point out some practical thoughts. The study is not providing any specific kits, so institutional supplies to be used for this, but that's where this particular specimen collection form specifies not only the quantities, but what kind of tubes to be utilized. So again, every site would have their own policy or process around it, but for example, I work at Mayo Clinic. This protocol was submitted to our biospecimen acquisition team or program, and based on this, we are just creating our own institutional kits that will come out to the patient's visits. That's just the standard way that we end up doing it. But just a reminder again that the study is not providing any kits for biospecimen collection. Institutions will be using their institutional supplies. Submission guidelines, as I said, it's to be sent to the SWOG biospecimen bank. You have the details. If you've participated in any other SWOG myeloma committee trials, the specimen submission guidelines are extremely standard as per that. Patient-reported outcomes. So these assessments, the EORTC, PRO, CTCAE, the QLQC30, they are to be done at baseline prior to registration, at months 1, 3, 9, and 18 after randomization. Each assessment has a window of plus minus 30 days of the target assessment. But obviously, certain things to keep in mind. If, for example, we are saying month 1 and 3, we don't want month 1 to be plus 30 and month 3 to be minus 30, because that would defeat the purpose. But other than that, there are some adverse event forms that are being submitted that will be collecting this patient-reported outcome, CTCAE, which I mentioned was fatigue, pain, and neuropathy. We are wanting to collect that data as the study proceeds. And then the PRO CTCA has a slightly different, as I mentioned, at baseline, at each treatment cycle, because the adverse event form will be submitted anyways, and that then at the discontinuation of treatment. We have assessed that these quality of life assessments will take about 10 to 15 minutes at every time point. So something to be able to convey to your patients, because of course we don't want the patients to be burdened, we don't want the sites to be burdened, but this is a very important part of the protocol. There are some details in the patient-reported outcome section about time points, how they are calculated, or what if a patient falls off somewhere in between an assessment, or what if one assessment is done and then the other assessment has not happened and the patient comes off trial in between, etc. All of that has been thought of and put in the section. So I would recommend, just for the research coordinators across the country, to please consider looking at that part, because that will help you clarify quite a bit of those questions. Follow-up, all patients will be followed for 10 years or until death, whichever occurs first. During the treatment period, follow-up is done obviously as per the treatment cycles. But if the patient is not on active treatment, then we're saying every 3 months in year 1, every 6 months in years 2 and 3, and then annually for up to the 10-year follow-up is complete or death. Of course, if the patient needs to be followed more frequently, then that could be as clinically indicated by the provider's discretion. As I mentioned, the funding for this, the daratumumab portion is being provided by the study. Bortezomib, len, dex will be used from commercial sourcing. There are some, the table and the time points for the study-related payments, etc. That is listed over here. A patient-friendly plain language trial summary and the accompanying social media toolkit are available. You have the plain language summary in both English and Spanish available in the room right at the back. Dana is kind of showing that to all of us. But they're also available through these links at the, they're available electronically also. And for those physicians who would like to use the social media toolkit, that has been prepared so that they can spread the word about the study. Another part for the EMR implementation, there is a template that is provided. And frankly, we've used this currently actually at Mayo. I know that portion is being done. And the feedback I've received from our kind of what's the Beacon build team is that that toolkit is very, very helpful. That gives them a very good, nice template to start with. All that they're doing is they're using that template. They are going to our internal the treatment plans for these regimens, and they're trying to figure out where the differences are, and that's where they're just asking the PIs for clarification if they need to. But they've at least given me the feedback that it's made their job much simpler. So please consider using those templates. I should also point out, right before I finish, that as I mentioned, all of us who are sitting over here, the SWOG team, we've been extremely passionate and focused on this particular study for the past few years that it's taken to come to this point, because as I mentioned, all of us myeloma treating physicians, and I'm pretty sure sites, feel that in general, this is a patient population that is always kind of excluded from clinical trials. So you noted that we have made quite a few changes to the inclusion criteria. I'm not going into those details, but there is actually a whole plan of racial, ethnic disparity mitigation processes or things that we are going to be doing on the side of this study. So there's actually that plan, which is the DEI, kind of the disparity mitigation plan, is hopefully going to be published very soon, even ahead of this trial, so that we want that plan to be widely available so that other trials can take some pointers from it and start using that. We know that there are these patients or the populations that don't get included in trials. Another extremely important point to remember is we got a buy-in and also excellent partnership from the VA for this study. So the study was just presented at the VA meeting about a week or so ago and is being activated at a lot of VAs across the country. So in our minds, that is also bringing the study as close to a real-world population as possible. Thanks a lot to obviously NCI for supporting these efforts, and also to Janssen for providing the drug and support, some other support for the study. You have a list of all the contacts for the study that are available also through the protocol and at the website. And I can tell you that so far our response time to emails has been in the matter of minutes to maybe less than hour. So we hope to be able to keep up that work. But I don't know, before I open up to questions, because I really wanted enough time for a dialogue, I don't know, Christine, Brea, any thoughts, anything that you want to highlight, point out? The studies, about 267 sites have been approved as of today by the NCI to open up the trial. We don't know how many sites have yet activated because we don't get to see that portion on our end at SWOG. But as I mentioned, kudos to Mercy Hospital in Des Moines, Iowa, because they've put on the first patient. Christine, anything else, Brea?
Well, it's very exciting.
Yep. So you have to turn that on. Yeah.
It's very
Hold it.
Thank you. It's very exciting to have the study open and the first enrollment. As Dr. Alwadi probably already highlighted that this study would, hopefully we can see most of the enrollment from community sites, and this is for the real world patient population. And so we look forward to the enrollment over the next couple of years and working with all the sites.
I just wanted to highlight one thing with this trial is that we all know that dexamethasone can be toxic for some of these patients. And so one of the things that this trial allows for is the discontinuation of dexamethasone, which is something that we'll be keeping an eye on as well. And another just point to bring up, something that we're doing at my institution where we've activated this trial, is we're using this sort of as an innovative way to reach out to our community partners and try and build new bridges and lines of communication so that our community partners who might not have this trial open can still have access to this trial for their patients. So it's been a unique way for us to collaborate. So I would encourage other people to think kind of outside the box and how we can reach these patients through our community sites. And as a referring center, if that's where you are, how can we be of assistance to these patients, as this is something that we do anyways. But if we can get them into trials, we can help standardize their care and make sure that we're delivering uniform care.
Excellent point. And so I should say that at the moment, so for this trial, the patients still do have to come to the treating institution, the enrolling site, to get all the treatment. Unfortunately, we're not at the point yet in the clinical trial machinery that patients can get local medical care because data collection, et cetera, becomes very tricky. But maybe we can imagine a future world somewhere where studies can be done in partnership like that between community and all kinds of sites. But as Christine and Brea mentioned, we are hoping a bulk of the enrollment to happen from community partner sites. That would be important. Any thoughts from anybody? Any questions? Any clarifications? You have please go ahead. Right in the front of the room. We can get you the mic.
Real quick, I don't know if I saw this, and if you did say it, remind me again what you're trying to accrue. What's your number?
Yes, it's the accrual target. So eligible patients are about 450, but with the, actually I did not mention that, and thanks for pointing that out. So the enrollment target, we are looking for about 450 eligible patients, but to account for the patients who may end up becoming ineligible after enrollment, so it's about 516, I believe.
Okay.
Yeah.
And then I did actually have another question.
Sure.
I know you said that treatment has to be done at like the sites,
Yeah.
but what about the patient-reported outcomes? Can they do that remotely? Can they do them from home?
So the patient-reported outcomes are set up for those time points when the patients ideally are supposed to be there for the visits.
Mm-hmm.
And that's one of the reasons we gave about that 30-day window, considering that even during maintenance, the patient would be coming back to the center once a month.
Mm-hmm.
Now, we don't have a way to capture the data. Now, these forms, these QOL measures are available kind of without licensing fee, et cetera. These are standard formats. but we don't have a way of capturing the data if the patient was to do it, for example, from home, online, et cetera. Then the data capture would not happen. We did bring up the question, and I'll ask maybe either the SWOG team or I don't know if Anshe, Adam, you guys have any idea about it, about whether these would be done electronically or paper format. And the way I understand is that it depends on how that particular site is set up, but most likely I'm thinking paper because then that data has to be submitted back to through RAVE or to the study. So my understanding is that it would be paper-based, but I do understand that some sites do it on electronically also. I should know, I do not know actually that if some site was to do it electronically, how the data capture happens. I don't know.
I mean, I think, and Adam, you may know better, but I think, I mean, eventually it needs to be in RAVE and electronic, right? And how the site actually administers that is up to the site. You know, it could be paper-based and then the CRA enters it, or it could be the, you know, somebody interviewing the patients while entering the data online. But I think that's up to the site.
Yeah, because eventually it has to come through RAVE to us. And I should mention that this was specifically entered into the protocol in advance because we really think this is important, especially for this patient population. And the kind of that payment grid, it does have site payments for those time points. We do understand that that would be added effort to the sites. So when we look at the budget grid, et cetera, there is a line item there.
Okay, thank you so much. I just wanted to say also, like, I'm one of the VA sites, and we're super excited to get this open.
Thanks a lot. I
Thanks.
really
Awesome.
appreciate that. We have another question, I guess.
Yeah, thank you very much for the summary. That was really good.
Sure.
A couple of questions. I think this one's straightforward. The categorization of frail versus intermediate fit, that's going to be like a stratification factor? Okay.
Yes, yes. So correct. So patients who start off with either of the frail or intermediate fit category, that will be one of the stratifications.
Okay, and then this is probably my interpretation, not understanding, but the PRO and the MRD groups, in the slides you presented, it was Arm A versus Arm 1 versus Arm 2 and 3 respectively. Does that mean 1 versus 2 and 1 versus 3, or 1 versus 2 plus 3?
So there is no plus in here.
Okay.
So we had looked at that and we did figure that the induction is the same between 2 and 3, but, and I'm trying to retrack in my mind the discussion. From what I remember from our last discussion is that the 3 arms are independent and there is
Okay.
no adding on to them.
Okay.
Even though that, because after the induction, arm 2 and 3 would split. So that's why the data analysis would become very tricky if we were to say that for induction time points, the 2 arms are clubbed together, and for maintenance, they would be separated. So I think we deliberately kept them separate, all 3. Adam is
Yeah.
Yeah,
nodding, so
that's
yes.
correct.
Okay.
You know, it's almost, you can almost see it as like 2 studies combined,
Yes.
right, in one. So it could be a study, Arm 1 versus Arm 2, where the induction is different, but the maintenance is the same. And another study where it's Arm 1 versus Arm 3, where both induction and maintenance are different. But we can answer both those questions in a single study without with fewer patients.
Yeah,
That's kind of how
Yeah, I think that's an excellent
about
question,
it.
and Anjay's explanation is spot on, because it took us almost maybe 6, 8, 12 months to keep hammering out back and forth, because whenever we would bring it up for discussion, the thought was, yeah, we need to compare VRD light and DRD, but what should be the maintenance? If you give LEN maintenance, yeah, that's deviating from standard of care for DRD. Or if you give DAR-LEN maintenance, then people would say, yeah, but we don't do that in real world. So then eventually we said, okay, if we really want to answer this question, we will have to create this 2 for kind of a study with 3 arms. It'll be a large patient population, but we got much better feedback from the SWOG community, and hence this design came up.
Okay, here's the last question. I think
Please. Go for it.
You explained it, but it was above my head.
No, please go for it.
This might be for the statisticians, too.
Sure.
This is an unusual trial where the primary and secondary endpoints are sort of split. Like, so, what is it? So the comparison of 1 versus 2 and 1 versus 3 for PFS and OS are, some of them are primary endpoint, primary objectives, some are secondary. What is the rationale for that?
That's a really good question. And it's again, you know, so 1 versus 2, that's PFS, and that's the one with the same maintenance, right? So the thought is, we want to get an answer as soon as possible, which we can get with progression-free survival.
Yes.
And because we don't change maintenance is the same in both arms, we can just, progression is a really good primary outcome. Versus if we are looking at changing both induction and maintenance in both arms, we really have to wait longer to
Right.
know whether there is a clinically meaningful difference in its overall survival. It's very confusing, I understand.
Yes.
But that's the, you know, again, if you think about it as 2 separate trials, then
Yeah.
it makes a little more sense, right? It's just 2 separate trials, but they're sharing the same control arm, right? The same
Right.
standard of care as arm 1. And then if we just did, you know, the arm 1 as a arm versus arm 2, PFS would be the outcome. Whereas if we change maintenance to, then overall survival would be the outcome. So it's just wrapped into, you know, this one
Maybe
trial.
I'll just clarify one quick piece before I'll ask you to please ask the qualifying question, is that when we said the primary objective, both of these that Anjay mentioned, PFS between 1 and 2 or OS between 1 and 3, they
Yes.
are both primary. This is a
Right.
dual primary, meaning
Yep.
any one of them comes back positive, the study is positive. But we didn't want to lose the opportunity of the other comparisons, meaning the other PFSs and OSs that are non-primary, so all of them were still
Yes.
made secondary. So the reason for that is, we're not gonna not compare any 2 arms. We're gonna do
That's right.
all sorts of comparisons, but the primary objective is 1 versus 2 and 1 versus 3. Please go ahead.
Right, and this is the last part of the same thought. Is it Angie? Angie, you mentioned that, and we all know this, OS is going to take a longer time versus PFS. So focusing on the primary endpoints, you're not going to get an answer to 1 versus 3 overall survival for quite a while.
You're absolutely right. That will take longer. But one of the main other thoughts was that since 2 factors. One, because this is a population in which truly that kind of a survival, long, just like the younger population, or the utilization of some of these newer strategies like CAR-T, bispecifics, et cetera, that in this population has not been reported, studied, and we don't even know how much impact it would have. Hopefully it will have a positive impact. That we may end up seeing some kind of an overall survival readout also, just because this is a special population. And also because there is the high-risk patients who are included in this, while they have been left out from, as I mentioned, that competing other trial, they specifically left out the high risk. So since there are high-risk and elderly frail patients, even with advanced kidney dysfunction, we may see something. So we didn't want to lose that opportunity. But you're absolutely right, the PFS readout may happen first.
Yeah, thank you. I didn't mean to hammer.
No,
you
that's an
guys.
excellent question. Those are all excellent
Thanks.
questions. I don't know if there's a way to see if any questions are present online, or I don't know if anybody can even submit. No, so no questions there. Oh, thanks. Any other thoughts from anybody, anything we can answer? But otherwise, we really look forward to everybody going back fully charged up, getting the study activated and putting patients on. Okay, perfect. Thank you. Thanks, everybody. Thanks for joining us.